Welcome to the deep dive. We're jumping straight into a scientific paper today, a real cornerstone that helped answer one of biology's biggest questions. How exactly does DNA copy itself? This is a classic from 1958 out of Arthur Kornberg's lab.
And, the absolute core finding, the really surprising bit, for the first time ever, they showed you could actually build new DNA in a test tube, outside a cell.
Wow.
Yeah, using an enzyme. But here's the kicker, maybe the counterintuitive part.
Yeah.
It only worked if they already had some DNA there to start with.
Ah, okay.
You needed that blueprint, that guide, couldn't make it totally from scratch.
Okay, let's maybe step back for a second and set the scene. Why was that such a huge deal back then? We're talking the mid-1950s, right?
Exactly. The double helix structure was out, Watson and Crick. Everyone was pretty sure DNA held the genetic code.
Right.
But the million-dollar question was how? How does this amazing molecule copy itself so perfectly every time a cell divides? That was the big mystery, the replication puzzle.
So people were hunting for the machinery.
For sure. Scientists were searching everywhere for the biochemical nuts and bolts. Arthur Kornberg's group, they were at Washington University then, they were laser focused on finding the enzyme responsible in E. coli, the gut bacterium.
Oh, okay.
And this paper, the one we're diving into, it's actually the second in a really key series. The first paper described how they purified the enzyme. They called it polymerase.
Right.
This second paper, published in the Journal of Biological Chemistry, digs into what that enzyme actually needs to do its job. What are the chemical requirements for making DNA? That was their goal.
So they've got this purified enzyme, they're suspect. What was the recipe then? What ingredients did they figure out were absolutely essential to get this DNA synthesis going in their test tubes?
Well, they were really systematic about it. They tested different combinations, and the results, especially if you look at Table I and Table II in the paper, they're crystal clear.
Okay.
The reaction had some non-negotiables. You absolutely needed highly polymerized DNA. They used DNA from cat thymus as their starting template, their blueprint.
Right, the guide DNA.
Exactly. You needed magnesium ions, Mg++ art, standard cofactor for many enzymes like this. And crucially, you needed all 4 of the deoxyribonucleoside triphosphates.
Those are the DNTPs, the
Yeah.
ATCNG building blocks.
The DATP, DGTP, DCTP, and DTTP, but in their high-energy triphosphate form, ready to be snapped into place.
And what happened if you, say, left one of those things out? What did the data show?
Oh, the tables show really stark results. I mean, dramatically different. If you left out the DNA template, synthesis just plummeted, basically 0.
Okay.
Same if you left out the magnesium. And here's a critical point. If you left out even one of the 4 DNTP building blocks, say no DATP, the reaction rate dropped by like hundreds of times.
Wow.
Table 2 used really sensitive radioactive labels to measure tiny amounts of synthesis, and it confirmed it. No significant reaction unless all 4 DNTPs were there together.
So it wasn't just adding random stuff, it was highly specific.
Exactly. It strongly hinted that the enzyme was following instructions using that template.
Okay, so they've got the ingredients list nailed down. But was the enzyme just adding maybe a few nucleotides here and there onto the existing DNA? Or were they actually making substantial amounts of new DNA, achieving what they called net synthesis?
Yeah, that's a crucial distinction, isn't it?
Right.
And that's exactly what Table 3 tackled. They let the reaction run for several hours.
Right.
And they measured the total amount of DNA afterwards using a few different methods, just to be sure. They looked at optical density, they measured the deoxy sugar component, and they tracked how much of the radioactive building blocks got incorporated.
Did the amount go up?
They absolutely did, significantly. The total amount of DNA increased anywhere from 2 times to even 20 times what they started with.
20 times.
Yeah. And importantly, they calculated that over 90% of the DNA present at the end actually came from those DNTP building blocks they added.
So it wasn't just tinkering, they were really churning out new DNA.
Genuine large-scale synthesis of new DNA molecules. Proven.
Okay, fantastic. They made new DNA. But, was it the right kind of DNA? Was it chemically correct? Did it have the same structure as natural DNA? How did they figure that out?
That's where they got clever with enzymes and chromatography. Figure 1 of the paper illustrates this beautifully. What they did was synthesize DNA using radioactive thymine, so trouble DTTP.
Okay, so the T's were radioactive.
Right. Then they took this newly made radioactive DNA and chopped it up into smaller pieces using an enzyme called deoxyribonucleic, or DNase. DNase basically just snips the DNA backbone.
Okay.
After chopping it up, they separated the resulting fragments using chromatography. Think of it like a molecular sieve running the mixture through a column where molecules separate based on properties like size and charge.
Sorting the bits and pieces.
Exactly. And figure 1 shows the results. When they analyzed the fragments from the digested, synthesized DNA, the radioactivity, remember, from the labeled thymine, showed up in very specific peaks on the chromatogram.
Not just smeared everywhere.
No, specific peaks. And these peaks corresponded to dinucleotides.
Meaning pairs. Two nucleotides stuck together.
Precisely. Pieces just 2 units long. And importantly, these dinucleotides contained the labeled thymine. Figure 1 specifically shows they found radioactive peaks for CT-citidylate linked to thymidylate and TT, 2 thymidylates linked together.
So that proved the radioactive T was actually chemically bonded to other nucleotides in short chains.
That's step 1, yes. It wasn't just floating free, it was incorporated.
And how did they know how those 2 nucleotides were linked? You know, DNA has that very specific 3' to 5' backbone linkage. Was it the right kind of link?
Excellent question. And that brings us to Figure 2 in Table 4. Figure 2 is basically a diagram showing how different types of enzymes cut nucleic acid chains at very specific locations or bonds.
Like specialized molecular scissors.
Exactly. So they took those radioactive dinucleotide peaks they isolated from Figure 1, the CT and TT fragments, and they treated these purified dinucleotides with a panel of different enzymes. Things like phosphodiesterase, 5-nucleotidase, phosphomonoesterase. Each one cuts a specific type of bond. Then they measured how much phosphate was released by each enzyme treatment. That's the data in Table 4.
Ah, so if a specific enzyme cut it and released phosphate, that told them what kind of bond must have been present.
Precisely. By seeing which scissors worked, they could deduce the structure, and their analysis confirmed it. The nucleotides in the DNA they synthesized were joined by the standard 3-foot, 5-foot phosphodiester bonds.
The exact same chemical linkage found in natural DNA.
The very same. So they weren't making some kind of, weird chemical analog. It was structurally authentic DNA.
Okay, so they proved thymine went in correctly, linked correctly. But what about the other bases? Did the enzyme incorporate A, C, and G properly too?
All right, they needed to show it wasn't just a thymine-specific thing. And that's the experiment behind Figure 3. This time, they were clever again. They synthesized DNA using radioactive labels on DATP, DCTP, and DGTP.
Huh?
They deliberately left out the label DTTP for this run.
Okay, so A, C, and G were hot this time.
Yep. Then they did the same procedure. Make the DNA, chop it with DNase, run it on the chromatography column.
And what did Figure 3 show?
It shows that same dinucleotide region of the chromatogram. And this time, they found radioactive peaks containing deoxycitolate C, deoxydedolate A, and deoxyguanylate G.
So radioactivity popped up in pairs containing A, C, and G, just as you'd expect if those building blocks were also being incorporated correctly.
Exactly right. So you take Figure 1, showing thymine incorporation, and Figure 3, showing A, C, and G incorporation, and together it provides really strong evidence that this polymerase enzyme was capable of incorporating all 4 types of deoxynucleotides into the growing DNA chains.
It really seems like they were systematically ticking off all the boxes to prove this was real DNA synthesis.
They were incredibly thorough.
Did they learn anything else about the actual process, the mechanism of how the enzyme adds those nucleotides?
They did, yes. Another really key observation is shown in Table V. They found that as the enzyme adds a nucleotide, taking it from a DNTP, like DTTT, and adding it to the chain, it releases something else.
Oh, what's that?
Inorganic pyrophosphate. PIPA, basically the last 2 phosphate groups from the triphosphate building block.
Ah, okay.
And Table V shows something really neat. The amount of this pyrophosphate released was pretty much exactly equal, mole for mole, to the amount of nucleotide that got incorporated into the DNA chain.
So one nucleotide goes in, one pyrophosphate comes out.
Precisely. This was a huge clue about the chemistry. It suggested the enzyme works by breaking that high-energy bond between the first and second phosphates on the incoming DNTP. That break releases energy and allows the remaining nucleotide monophosphate to form the phosphodiester bond with the growing chain, releasing the PIPA.
That makes sense chemically. Now, didn't you mention earlier that high levels of this pyrophosphate, the PB-PUN, could actually slow down or inhibit the synthesis?
I did, yes.
Does that imply the reaction could potentially go backwards? Which feels a bit weird for synthesis, right? You want it to go forwards.
It does seem a bit counterintuitive, but that's exactly what they investigated with the pyrophosphate exchange experiments. These are detailed in Tables 6, 7, and 8.
Okay.
They confirm that yes, while lots of people could inhibit forward synthesis, the enzyme could also catalyze an exchange reaction. They used radioactively labeled PPA, P-labeled pyrophosphate.
Right.
And they showed that this labeled PPA could actually get incorporated back into the pool of deoxynucleoside triphosphates, DNTPs. And crucially, Table 6 shows this exchange reaction absolutely required the presence of the DNA template.
So the enzyme sitting on the DNA could essentially swap a labeled PPA floating around for the unlabeled one attached to a free DNTP.
That seems to be what's happening, yes. And Tables 7 adds another layer. They compared what happened when they left out individual DNTPs. We already know that leaving out a DNTP basically stops synthesis dead.
Right, drops by hundreds of times.
But interestingly, leaving out one DNTP only moderately reduced this pyrophosphate exchange reaction.
Huh. So even if it couldn't build the chain because a block was missing?
It could still seemingly interact with the DNA and the available DNTPs and carry out this exchange. It suggested the enzyme was still active in some sense at the template, even if polymerization wasn't happening efficiently.
Did this exchange work with the DNA they made in the lab, too?
Good question, yes. Table 8 shows the exchange reaction occurred even when they used the enzymatically synthesized DNA as the template, not just the original Cap-finus DNA. So it wasn't some weird artifact of the starting material.
This reversibility, this exchange, must have given them a really deep insight into the chemistry, didn't it?
Absolutely.
Yeah.
It strongly reinforced the idea that the fundamental chemical stack was breaking that triphosphate to release PITA and form the phosphodiester bond, and that this step, chemically, could be reversed under the right conditions.
Right.
They even speculated in the paper that maybe very short, newly added bits of the DNA chain might be more susceptible to being snipped off again by this reverse reaction pyrophosphorolysis, they called it, compared to nucleotides within a longer, more stable double helix.
Okay, we've really walked through the experiments now. If we pull back a bit, what were the big conclusions, the main hypotheses that the authors themselves drew from all this amazing data in their discussion section?
Well, they synthesized it really powerfully. A huge one was how they interpreted that absolute need for pre-existing DNA.
The template requirement.
Yes. Their data strongly suggested it wasn't just acting as a primer, a starting point for the enzyme to add on to. They proposed it was also, crucially, acting as a template.
Guiding the sequence.
Exactly. Guiding the synthesis of a new complementary strand based on the sequence of the original DNA. This was a really groundbreaking hypothesis at the time, and it fit perfectly with the Watson-Crick model, suggesting how DNA might be able to duplicate itself.
So their biochemistry provided real experimental weight to that structural model.
It really did.
Yeah.
They also emphasized the enzyme specificity. It clearly preferred deoxynucleoside triphosphates. It wouldn't use diphosphates, and importantly, it wouldn't use ribonucleotides like ATP, the building blocks for RNA.
So it was definitely a DNA polymerase.
For sure. And they noted something else interesting, too. While really harsh treatment with DNase obviously destroyed the template DNA,
right,
they found that mild DNase treatment, or using a particular enzyme fraction from E. coli, could actually activate the DNA template, making it work better in the synthesis reaction.
Huh. Why would that be?
Well, they didn't know for sure, but it hinted that maybe the physical state of the DNA mattered. Perhaps the enzyme preferred templates with nicks or ends to start synthesis from. Interesting little wrinkle.
This whole body of work just feels so foundational. How did it really change the game in molecular biology, and what's its lasting impact today?
Oh, the impact is immense. It resonates all through modern biology. First off, the methods they developed were revolutionary in themselves.
How so?
Their basic quantitative assay for measuring DNA synthesis using radioactive nucleotides and then precipitating the DNA with acid to separate it from unused precursors, then measuring the radioactivity incorporated. That became the standard method for studying DNA polymerases for decades.
So they didn't just make the discovery, they gave people the tools to study it further.
Absolutely. It provided a reliable quantitative handle on the process. Then, beyond the method, they fundamentally established the core chemical requirements. All 4 DNTPs, magnesium ions, and that crucial primer template DNA. These are like bedrock principles we teach day 1 in molecular biology now. And their findings provided essential biochemical evidence that strongly supported the models of DNA replication that were emerging, particularly the semi-conservative model that Messelson and Stahl would famously prove shortly after. It also paved the way for understanding later discoveries, like how accurately polymerases copy DNA fidelity and the existence of proofreading mechanisms that fix errors.
And you have to mention the connection to PCR, right? That seems like a direct descendant.
It absolutely is. The fundamental reaction they described in this 1958 paper, an enzyme using a DNA template and DNTPs to synthesize a new DNA strand that is the absolute core principle of the polymerase chain reaction, PCR.
Which is everywhere now.
Everywhere. Developed decades later, of course, but PCR relies entirely on the basic enzymatic activity Kornberg's lab characterized here. It's used in medical diagnostics, genetics, forensics, all kinds of research. It's a direct technological legacy of this fundamental work.
It really is incredible, that straight line from basic biochemistry in the 50s to a technology that impacts almost everyone today.
It's a perfect example of how curiosity-driven fundamental research can unlock completely unforeseen applications down the road. And, the importance wasn't missed at the time. Arthur Kornberg won the Nobel Prize in Physiology or Medicine in 1959, just a year after this paper came out, specifically for this work on enzymatic DNA synthesis. It's rightly considered a giant classic in the field.
So zooming out for everyone listening, why should you care about a paper from way back in 1958? Well, this work on DNA polymerase isn't just some dusty historical artifact. It explains the fundamental chemistry behind how the genetic blueprint in your own cells gets copied faithfully billions of times throughout your life. It's essential for growth, for healing, for life itself. And as we just heard, the principles uncovered here are the engine-driving technologies that are critical in medicine, in forensics, and understanding our world today. It has very real, tangible impacts.
And, even with all these groundbreaking discoveries, it's interesting that the authors themselves mentioned in the paper's discussion that they hadn't yet figured out what ultimately limited the synthesis. Yeah, what factors govern the sort of ultimate synthetic potential? How long could the enzyme keep going? What eventually stopped it?
So even in this landmark paper, they were pointing out the next set of questions.
Exactly. Even milestone discoveries always leave fascinating new questions unanswered, paving the way for future research. It's how science keeps moving forward.
That's a fantastic thought to end on. What an amazing journey through this classic paper. Thanks for unpacking it with us.
My pleasure. It's a great one.
And thank you for listening to The Deep Dive. Keep exploring.